Hello, my name is Isaiah George, and I am a microbiology major and a Texas Tech Red Raider. As a Sizer SSO research scholar, I conduct research in the areas of chronic wounds, biofilms, and clinical microbiology. Under the mentorship of Dr. Allie Smith, our research lab operates in the Department of Surgery at TTU-HSC in Lubbock. In this video, I will give you a quick tour of the Smith lab and also show you a neat bacterial fluorescent imaging device that we use in some of our projects. When you open the door to our lab space, you will notice straight ahead another doorway. This is where we keep our autoclave, double distilled water, and our -80 cryo freezer. For more information on these, please visit the lab tour video given by Rachel Diaz, who is another member of Smith Lab and also a Sizer scholar. When you look to the left, you will see our workbenches and also the hand washing station right in front of your eyes. Now, keep in mind that our lab space is very old. In fact, this very room used to be part of the hospital rooms for patients a very long time ago. This overview image of our lab, as well as some of the other equipment in our lab, all shine light on the old, yet long established and well-functioning equipment that keeps our lab running. Here's another overview image of our lab, with our workbenches to the left and right, and our incubators straight ahead. This is our drying incubator, and this is where we place things like glassware and pipette tips that just came out of the autoclave. We also will place augers that just came out of the fridge into this incubator as well. Microorganisms thrive in moist and humid conditions, so it is important to ensure that materials are properly dried before putting them to use. This is our sterile incubator, which means that instead of things like glassware and augers, we store our bacterial cultures in here. This incubator is intended only for culture plates, and the temperature is kept around 37 degrees Celsius. Since we care about bacteria that affect our human bodies, studying these bacteria include growing cultures at the temperature of the human body, which is 37 degrees Celsius. Here we have our fridges. In the far left fridge is where we store our bacteria cultures and plates after they have been processed from the sterile incubator. The middle and right fridges hold our augers and other items needed for experiments. In this image, the black shelves in the back hold our auger and broth media, as well as other disposable lab equipment that we frequently use. You can also see our dishwashing station here. This is where we clean glassware and reusable containers after autoclaving. This is our shaker. This machine is set to a temperature of 37 degrees Celsius and essentially shakes the contents inside at any chosen circular speed in rotations per minute, or RPM. Right now, we are growing in vitro biofilms inside. A biofilm is basically a film or community of microorganisms, usually more than one species, all of which are encased in a slimy extracellular matrix. Let's take a closer look. In this glass tube, you can see a scratched pipette tip. This acts as a scaffold for the biofilm to grow on and attach to. And here is another image. These biofilms are all monomicrobial, or one species, with just Staph aureus. If you look closely, most of the biofilm in this glass tube is contained in the red circle and attached to the pipette tip. Now I want to show you the basics of a bacterial imaging device that we use to detect bacteria, but not just any bacteria. This handheld imaging device detects porphyrin-producing species only, which is important to understanding the way the device works. Keep in mind that a large number of pathogenic bacterial species produce porphyrin anyway. In this image, we have blood agar and PTA, both of which are cultured with the same porphyrin-producing bacterial species. In this image, you can see the MolecuLight handheld imaging device that detects the autofluorescent properties of porphyrin-producing bacteria. If you look directly at the Petri plates, you can see that the bacteria on the PTA plate are fluorescing red, whereas that on the BAP plate are not. The device emits a violet light, and the device camera captures the red fluorescence produced by these porphyrin-producing bacteria. Here is another close-up image of the two plates. And here is a close-up of what we see on the screen of the MolecuLight device. Now, we have this cool device, but you may be wondering why it is important. The application of this device serves useful in detecting infectious bacteria and chronic wounds. Many chronic wounds harbor biofilms, which make the wound more difficult to treat. The biofilm acts as a mechanical barrier, if you will. One application of this device is to help a physician locate where exactly in the chronic wound infectious bacteria are residing so that a faster, more effective treatment can be administered. This is an easy to work with device and one that we work with quite often in our projects. And that wraps up the Smith Lab tour. I would like to acknowledge Sizer SSO for the sponsoring of this video and their support in our research. I would also like to thank TTUHSC and Texas Tech for their support in providing a lab space. And I would also like to thank MolecuLight for their collaboration in our research endeavors. And lastly, I would like to thank Dr. Smith and all members of Smith Lab for their dedication to research and support for one another in making our lab the success that it is. If you enjoy this lab tour or have any questions regarding our lab, please reach out to Sizer SSO and we will get in touch. Thank you for watching.